So maybe you've heard this before. The human body is 2/3 water. Seriously? Where is this water? What's it doing? And why don't we slosh when we walk? Welcome to Pocket Science, a compact guide to the human body and how it works, powered by the world-class scientists of the Fralin Biomedical Research Institute at VTC. I'm your host, Matt Chittum, and today we're going to ask that question to Jenny Munson. Jenny's an associate professor here at the Research Institute. She's a researcher, tissue engineer, and an expert in fluid flow in the human body. So Jenny, welcome to the podcast.
Thanks for having me.
I don't know what you think of me, but I tend to think of myself as a pretty solid guy. But is this true? Am I mostly liquid?
Yeah, you are mostly liquid. Fortunately, about 2/3 of that liquid is housed inside of cells in your body, so kind of the smallest biological unit that's making up your body. So it's not just sloshing around, it's inside of something that then is contributing to form within your body. And then that other third of all of that liquid is going to be outside of those cells, and it's going to be still contained within different structures or features or other things that hold your body together in different ways.
Got you. So that makes sense. And I think where you might be going with this is something called those spaces in between, the interstitial fluid flow. Until we'd met, I'd never thought about the spaces in between the sort of solid things in our body. But that's your realm. So tell us more about interstitial fluid flow and how it works and all of that.
Yeah, so I think it's pretty normal to think about fluids in your body that are moving in the tubes in your body, things like blood, which all of us have seen when we go to the doctor, and hopefully that's about it. But you're going to have all this other fluid that's moving in your body, and some of that is also in tubes, things such as lymph, which move through your lymphatic system, which is really important for draining waste from your body. But then you have the fluid that comes between the blood and the lymph, and that's going to be your interstitial fluid. And these fluids are really important because they're really bathing all of those cells and all of those tissues that are kind of functioning on a day-to-day basis. So that needs to be constantly moving and replenished and cleaned so that you have healthy function in every single one of your organs.
Okay. Pause right there. Jenny's very matter-of-fact about this, but it's really kind of crazy. This interstitial fluid, interstitial means between things, by the way, is all over your body. And it's not just water. It's sugar, salt, hormones, neurotransmitters, and each of us has about 11 liters of it. That's 3 gallons. For context, that's more than twice as much blood as we have. Okay, back to the conversation. Lots of labs are focused on a particular disease or something like that. But in your lab, you've studied this process in the body, if that's the right word, this fluid flow. People often, if they know your work, they think of you as maybe a cancer researcher, but you're really taking that understanding of how the body works and applying it to not just cancer, but lots of different diseases. Can you talk about the different things that you're working on in your lab in that regard?
Yeah, that's right. We started out in cancer mostly just because of opportunity and interest and, the area that was really hot to study when I first started this some 15 to 20 years ago. But these fluid flows, these interstitial flows, they've been studied for decades across the body, and there's some universal truths to them and how they move and how they cause different cells and tissues to change and function, both across different tissues, such as we look in the brain, we look in connective tissue or in adipose tissue or in pancreatic tissue, both in kind of a normal state, but also during disease such as cancer, but also in Alzheimer's disease, in diseases such as lipedema, which is a form of drainage disorder that can occur in the extremities of tissue, in your extremities. And, just kind of in a lot of applications that come up through, great collaborations that we have formed over the past decade and a half or so. And so we know that these flows, these interstitial flows are happening, in normal states and disease states, but fundamentally they're all kind of driven by some similar processes and kind of are beholden to certain rules within the body and within the tissues that can really affect their rates or their directions. And that's what really excites us in my lab, is kind of trying to find these universalities across the body, across diseases, so that maybe we can translate our findings from one thing to another.
So talk about fluid flow. I know you do a lot of work on glioblastoma, which is a highly lethal brain cancer. Talk about fluid flow in that context. What have you learned about how fluid flow works in that kind of cancer, and what can you do with that knowledge?
Yeah, so in brain cancer and in most cancers, solid cancers, so cancers that form an actual tumor in a tissue, what happens is as that tumor forms, it creates a heightened pressure in that tissue. And that tumor has a very high pressure because it's growing unchecked within a confined space. And then, but around that tumor, everything else is relatively normal compared to the tumor itself. So you have a normal pressure, which is lower than the tumor pressure. And so the analogy I like to use is that like a weather system where you have kind of a high pressure system next to a low pressure system, and that causes kind of wind to blow towards a low pressure system, this high pressure tumor next to the normal tissue will actually drive fluid from the tumor into the surrounding tissue. And so that can have a lot of effects. One, that flow rate, the speed at which that flow is moving is much higher, and that can cause tumor cells to invade away from the primary tumor and into the surrounding space. And that's what we've really studied for the past 15 years or so. And we know that we don't want that because it makes the tumor harder to cut out, harder to treat, harder to target.
Wait, did you get that? This fluid flow could be part of the problem that makes this terrible cancer so hard to beat. This is the cancer that killed Senator John McCain and President Joe Biden's son, Beau. Few people survive more than a year and a half with it. What Jenny's saying is that this accelerated fluid flow could be spreading the tumor cells into the surrounding brain tissue. So when the tumor is removed surgically, it leaves behind little bits of itself to start growing again. Okay, back to Jenny.
But it can also cause a lot of changes to the tissues surrounding those tumors. It can cause changes to the cells that are normally there that can aid the tumor or can cause a host of other effects that we're really trying to understand on a fundamental level. And what that all means is that if we can do something to either normalize that flow, redirect that flow, or even leverage what we know about that flow and what that flow does to use different drugs or target drugs differently, or potentially just to inform where we want to actually target a treatment, that could really help in treating this disease.
You mentioned Alzheimer's disease. Is fluid flow different in that disease?
Yeah, so that's what's, I think, really interesting about the nature of these flows is that there's no one size fits all. So in cancer, we see heightened flows, so the flows are much faster, and that's true in the brain, but it's also true in breast cancers. It's true in other solid tumors. In Alzheimer's disease, you see a slowing down of flow. And so in patients as well as in other models, we see that flow slows down drastically with Alzheimer's disease. And actually, if you can potentially right that flow and speed it back up to a normal rate, then you can kind of alleviate the effects of that disease and get better cognition.
Jenny, I know another part of the work that you do in your lab is tissue engineering. You're an engineer by training. Talk about that part of your work.
Yeah, so tissue engineering is, for those that might not know, is the building of tissues in a lab or even, in a body, but we do this just in the lab. And tissues are going to be another kind of fundamental unit of your body. And so they're going to be a collection of cells as well as the stuff that holds the cells together, which is called extracellular matrix. And then there's going to be a lot of water there. And so what we can do is actually take the cells and then a matrix and then put them together. Cells and gels is what we often just say, kind of offhand. And then that will create a tissue. And this is something you can actually pick up, sometimes with your fingers. You shouldn't without a glove. And then you can handle it and you can manipulate it. And it's an actual component as opposed to a cell, which you would really need a microscope to see.
Okay, last time, promise. Think about this. What Jenny's describing is engineering a model of a real individual patient's tumor microenvironment. using the patient's actual cells. And what this means is potentially doctors could then test different treatments on that model rather than the patient to determine a personalized treatment for their cancer without doing all the trial and error on the patients themselves. So we're talking about one day being able to start cancer treatment way down the line of knowing what's more likely to work for each individual patient.
And so what we use these for and why we need them is that we're studying interstitial flow. And interstitial flow is moving through the interstitial space, which is the space between the cells. And so if you don't have this kind of 3D system through which these flows can move, you can't really understand how those flows affect the cells in that environment. And so that's what we really use these for. And so this is something we do on a regular basis to model all sorts of diseases, which we've kind of already mentioned, or such as cancer, Alzheimer's disease, lipedema, breast cancer, things like that.
Talk about one particular application of that. I know you do that with regard to, I think, glioblastoma. Is that correct?
Yeah, so in glioblastoma, which was really one of the first tissue-engineered models we developed, we used these systems to apply different interstitial flows and then look at how these cells, the glioma cells, will actually invade. And invasion is the thing that you want to stop when you're talking about glioblastoma because it does make it very difficult to treat and causes recurrence. And so that's what we have built, and then we can add other cell types in there, other proteins or other types of factors that might really affect how these glioma cells respond to the flow and also just to the surrounding environment in which they live. And what's really interesting with these models is that when we do this and we take cells that we have from different patients that are personalized, we can actually see that not all of these cells respond the same to fluid flow. And so in that way, we're able to start to understand how we might be getting kind of personalized responses to fluid flow and how we might be able to pick or choose different drugs and personalize the medicine for those patients based on how these kind of tissue-engineered models respond to different treatments and to flow and to other types of factors that might be surrounding those cells.
Fantastic. Thank you for that. So it just seems like you're going to a lot of really brand new places with this research with lots of possibilities. What's the potential power of what you and your team are learning and discovering? And when you think about where all this is going and the impact it could have, what do you think about it? What keeps you going?
Yeah, so I think one, as a scientist at my foundation, I'm just curious and just interested in understanding what this flow is and what's driving it and how can we change it. So I'm really excited about that side of things just because that is kind of what got me into this game to start, is that I had questions and I want to answer them. But then, to really drive home what we can do and kind of the vision of where we could go, thinking about how it could actually affect, patients and individuals is what drives, getting into the lab or to my computer every day to try to understand, how what we know about flow can be applied in a real world setting. And I think as an engineer, that's why I kind of apply that approach to things, because engineers, we're always looking for solutions. We want to solve problems. And so, those problems that we can solve, we see as being using our expertise in how fluids move and how they might move across the body and applying that to a patient who might need it, whether that be for helping them, better target a tumor, to maybe alleviating the effects of chemotherapy, or, that might cause things like lymphedema, or by really, just fundamentally understanding and validating and finding new ways to study diseases that are lesser known, like lipedema.
Fantastic. Thank you so much for being here, for walking across the street to be with us. Ladies and gentlemen, Jenny Munson, Associate Professor at the Fralin Biomedical Research Institute. That's it for this episode of Pocket Science. If you like what you heard, follow us and subscribe to Pocket Science on your favorite podcast platform. Or find us on the Fralin Biomedical Research Institute website, fbri.vt.edu. Pocket Science is produced and engineered by Clayton Metz, and I'm your host, Matt Chittum. Till we meet again, see ya.